90% of the 2025 NECO Biology practical examination questions have been thoroughly answered in this video. Hello and welcome to my channel. In this video lesson, I've analyzed the various specimens for the 2025 NECO Biology practical examination, and I've also answered likely questions that you'll be given on the day of the examination. So do well to stick around, get your writing materials, don't skip any part of this video, and watch this video lesson to the end. Before we look at the various specimens, here are certain tips you ought to know, and these tips will enable you to perform better in the biology practical examination. And the first is that the practical examination makes up about 35% of your final score. And this simply means that doing well in the practical examination will give you an edge in getting a good grade. Now, what does this mean? You need at least 50% to pass any subject in the NECO SSCE, and 50% is equal to a C6. So imagine getting 25% from this practical examination, and then the other 25 from the objective and the theory. That is equal to 50%, and with that you have passed the biology practical or the biology examination as a whole. So I'm encouraging you to do very well in the practical examination, and you should take it as serious as possible. Secondly, when you are asked to give the scientific name of any organism, which they will definitely ask you to do, the genus name, that's the first name, should always start with a capital letter. So the scientific name for maize is Z maize, and as you can see, the Z starts with a capital letter, and the second name, which is the specific name or the species name, should always start with a small letter, and then you should underline each of them separately. So because these words, they are not English words, so remember, start the first name with a capital letter and the second name with a small letter, and thereafter underline them separately. Thirdly, you should not paint or shade. As I always say, in biology, it is not fine art, so they don't expect you to draw something as exact or as beautiful. Your diagram should just be something that should represent whatever you have been given. So your diagram should not be as they don't expect it to be as beautiful or as very colorful. So don't paint or don't shade, because if you do so, you'll be penalized for doing so. Just draw to your own ability and label properly. Fourthly, when you are told to label, or definitely you certainly label after drawing. So when you want to label your work, label or your diagrams to one side. So don't label in a haphazard or in a random manner. So if you are taking your label to the right, just simply do so to the right. If it's to the left, just simply maintain all your label to the left. And also, lastly, when you are told to contrast means what's the difference. So they can tell you to contrast or find the difference. When you are told to do so, simply draw a table. I'll show you in the subsequent sections of this video. So these are important tips you ought to know, and if you want to know better on certain things you ought to know, the do's and don'ts of biology practical examination, you can check up on this video showing right now on the screen. So moving on, here are a list of specimens for the 2025 NECO Biology practical examination. Now you have from Specimen A to Specimen O. So one advice I'll give to you as a student, after watching this video, simply try to go through the specimens again, and also try to master the diagrams of some specimens, because in biology you must be told to draw. So master their diagram and also master their labels much as possible. So the practical examination is just a framework, because whatever you have been given, you certainly see them on the day of your examination. So there are certain questions that they must certainly ask you. For example, if they should bring a question on the housefly, they must tell you to identify. They will tell you to give the scientific name, they will tell you to give the economic importance and certain questions. So it's just a framework that you ought not to play with. So Specimen A is the housefly, which we'll look at very soon. Specimen B is spider, C is the pride of Barbados flower, D is the bean seed, E is butterfly, F is crayfish, G is grasshopper, H is the humerus of a rabbit, I is the femur of a rabbit, J is an orange fruit, K is the honey bee, L is the land snail, M is the guava fruit, N is the Irish potato tuber, and lastly specimen O is the sweet potato tuber. So our first specimen is the housefly, and if you look towards your left, we have a diagram of the housefly. So in your exam, you could be told to pin the housefly on a table and then draw and label. So this is the housefly. On the housefly, there are certain parts you must know. For example, we have the antenna as indicated here, we have the tongue, we have this whole structure here which is the head, we have the first leg, we have the second leg, and then we have the third leg. Then the housefly has a compound eye. Then the part below the head, that's this region, is called the thorax. We have the wings. Okay, I've talked about the third leg, and then we have the leg has two parts called the femur, and then beneath is the tibia, and then we have the tarsus, and then the foot. So this is a diagram of the housefly. So I will beg you to master this diagram and also part of the housefly. So when you are done, it's always good to give your work a title. So you'll name this as the dorsal part of a housefly. So that's the title for this diagram. So what are the questions they can ask you? The first is to identify the specimen. So in your exam, when they tell you to identify a specimen, it simply means what is the name of that specimen. So in that space, you simply write housefly. Also, they can ask you for the scientific name, and the scientific name for the housefly is called the Musca domestica. And if you look at my writing, Musca, M starts with a capital letter, then domestica, D starts with a small letter, and then they are both underlined separately. So in your exam, they can go further and ask you to tell them the kingdom of this specimen. The housefly is from the kingdom Animalia. It is from the phylum Arthropoda, and it's from the class Insecta because it's an insect. And the order is what we call the Diphthera. Diphthera, this di means two. They are insects with two wings, so that's why the housefly belong to this order called Diphthera. So the next question they will possibly ask you is to tell them the features of specimen A. Now features can just be what you know. They can also have to list observable features. Observable features are what you can see right there. So what can you see looking at this specimen? There's the head, so you can write presence of head. There's the thorax, presence of thorax. There's the abdomen, so you have three parts: the head, the thorax, and then the abdomen. So presence of the abdomen. Then there's a pair of antenna. 1, 2, so that's a pair of antenna. You have three pairs of walking leg. You have the first leg up, you have the second leg, and then you have the third leg. So you have three pairs of walking leg, and then presence of two pairs of wing. Then they can also ask you to talk about the feeding habit. How does this animal feed? It feeds through a means called lapping and sponging. So majorly they feed on decaying materials or even good substances. So for example, they can feed on physics and other substances, or even good food like somebody's meal that the person is eating. So they do that by what is known as lapping and sponging. They don't bite, so they do what we call lapping and sponging. So they can also ask you the type of metamorphosis. So what's metamorphosis? Is simply the changes an insect will undergo before it gets to its final, or an animal will undergo from the egg stage to the adult stage. So for the housefly, it undergoes what we call complete metamorphosis. And they can also talk about complete metamorphosis. So in complete metamorphosis, the insect undergoes 4 life stages, which is from the egg to the larva, to the pupa, and then to the adult. So take note of that, the type of metamorphosis. Next, they could ask you to talk about the economic importance. Now, when most students see this particular question, they will think because of the word important, it means talk about the good side. No, economic importance means both the good and the bad side. So any good and bad you can remember about that specimen, you are free to write it down. So for specimen A, it's a vector of diseases because it carries parasites that causes several diseases like cholera, typhoid, and dysentery, and the rest. Also, they can contaminate food, and also they can reduce agricultural activity. So also they can bring about a loss in economic food industry. Imagine just cooking your food and you see a lot of fly on it. You simply throw away that food if you don't want to contact certain diseases. So that could bring about a loss in food industry. Also, the larva can be of help because they can help to decompose organic waste. So it's very, very important as the larva is used for decomposition. So the last question they will possibly ask you to talk about the method of prevention. How do we prevent housefly? So number one, you can use insecticide or pesticide. Secondly, you can do what we call proper sanitation by keeping your environment clean and everything around you clean to prevent them from coming close. Also, cover your food and water so that they don't come in contact with it. Also, regular clean of drains and water to ensure that whatever will bring them close to you is out. Also, proper disposal of waste products and avoiding defecation. That's open defecation means just going to an open site and just passing feces out. And also keep one toilet clean, because if they are not clean, it will attract the housefly. And lastly, destroy their breeding site. So our second specimen is the spider. Again, they could try to identify means what is the name of specimen B. That's the spider. So you are simply going to write spider. The next question they will ask you to tell them the scientific name, which is the Arenae species. And for the classification, I've given it over there. It's similar to that for housefly, but the class here, the class of the spider is the Arachnida. For the features, what you can see, so the body has what we call a cephalothorax. Now, a cephalothorax is simply the head and the thorax fused together. So here they don't have a distinct or a separate head, unlike the housefly that has a head and a thorax. Thorax just simply means chest, but for this species, the head and thorax is joined together. So that's why we have the name cephalo. Cephalo means head, thorax means chest. So they are fused together. So they have eight legs, as you can see on the diagram. There's no antenna. They have what we call a simple eyes, and then they have spinneret for making webs. So most of us are familiar with spider's web. That spider's web is made from this structure at the back called the spinneret. So this is a diagram, so it's very important to master the diagram and also the parts so that you can properly label your work on the day of the examination. Again, this is the dorsal view of the spider. For the feeding habit, it is carnivorous and it can feed on insects that it finds on its way. Then for metamorphosis, it undergoes incomplete. Remember, housefly undergoes complete. So for this incomplete, they go from egg to what we call spiderlings, then to the adult spider. So for incomplete metamorphosis, they have 3 stages, while for complete, just like that for the housefly, they have 4 stages. Then the economic importance. Number one, they act as natural pests. Remember, we said they are carnivorous. So for this reason, they can feed on other insects and also help to control pests in their environment. So they can be used for scientific studies. And lastly, their wool or their web can be used for production of silk, which is a clothing material. Next, they can ask you to talk about their method of prevention. How to prevent them? So one thing is to clean hidden corners, because obviously that's where spiders, they build their webs. Also silk crack, because that's a good spot for them to also build or create their web. Also remove webs, which is their houses, and then use sticky traps or insects. This will help to make them stick to it, and then you eliminate them. Now, this is a very good question you definitely see in the exam. They will try to talk about the similarities between housefly and spider. So when they say similarities, just keep using the word both. So in this case, both of them are arthropods, because arthropod means they have jointed legs. So, sorry about the second point, but you can also write it on your examination. So they both have jointed leg. Next, both have what we call external skeleton, and that external skeleton is what we call an exoskeleton, and their body are segmented. So let me show you the segmented body with the housefly. You see a line here, this a line, this and that line, this and that line. Though it's not obvious on the diagram we have for spider, but segment just simply means division. So the last question that we ask you from A and B is to tell them the differences. Remember, I told you that when you are told to talk about differences or contrast, always draw a table. It's very important. If you don't do that, you are at the verge of losing marks. So for housefly, they have 3 pairs of working leg, while for spider, they have 4 pairs of working leg. You see it on the picture. Then housefly have antenna, while spider do not. Housefly have compound eyes, but spider have simple eyes. Next, housefly have wings, but spider do not have wing. And housefly, they belong to the class of insecta, but spider belong to the class of arachnida. Specimen C is the pride of Barbados flower, and this is a very common flower in most of our neighborhood. So this is the flower shown on your screen. And moving on, this is the diagram of the pride of Barbados flower. In your exam, you most likely be told to cut this flower in a longitudinal section. Longitudinal means from top to down. You will be told to use a razor blade to cut the flower from top to down. So when you do so, this is what you are going to get. So in drawing, I think you should to draw something good, and then you will then be told to label. So if you look at the diagram, we have this colorful part, that's the red part, which is called the petal. So it is a brightly colored part. Then we have this anther above, where the pollen grains are produced. Then you have the filament. The filament is this line that holds the anther in position. So both of them mix up what we call the stamen. Next, and this is the male part of the flower. So the stamen is the male part of the flower. Then we have the pollen. Okay, above we have the stigma on top. Then there's a tube that runs from the stigma down. This tube is called the pollen tube. And then we have the style. So we have the stigma, we have the style. Within the style or inside the style, you have a pollen tube running down. And lastly, you have this ovary where the ovules are stored or are kept. Now altogether, this entire part, the stigma, the ovary and the style, makes up the pistil. And the pistil is the female reproductive part of this flower. So you have the ovule, which are the reproductive structures. Then you have this brightly colored part called the petal. You have the receptacle, which is the green part that is like a leaf just below this colorful part. Then we have this swollen part called the receptacle. And lastly, there's a stalk that holds the flower to the stem or to the plant. That stalk is called the pedicel. So in your exam, they will tell you to draw the longitudinal section of the pride of Barbados flower. So I've carefully named that so that you can do so on the day of your examination. So moving on, they will tell you to identify. When you are told to identify, simply means what is the name of the specimen you can see. So Specimen C is the pride of Barbados flower. The next question you'll be told to answer is, tell them about the scientific name, which is the Sassafina pusherrima. Again, C is in capital letter, P is in small letter, and they are underlined separately. Then for the kingdom, it belongs to the plant kingdom, and the division, it belongs to the Tracheophyta. And for the class, it's a dicotyledon because it has 2 seed leaves. Next, for the observable feature, what can you see on that specimen? So the petals are brightly colored. That's why it's indicated as brightly colored petals. Also, the appearance of sepals, that's like the green part below the petals. You have stamen, the male reproductive part. Also, you have pistil, the female reproductive part. Also, there's the presence of pedicel. The pedicel helps to attach it. The pedicel is the stalk I told you about. This is the pedicel. It attaches the flower to the parent plant, and also their compound leaves. Next is the function. You'll be asked this question. What are the functions? It attracts pollinators, that's agent of pollination like insects. Also, it is for reproduction. So in reproduction, that will lead me to pollination. So what's pollination? So pollination is simply the transfer of pollen grain from the anther to the stigma. So I'll come back to the diagram. If you look at this diagram, this is the anther. This anther produces a male gamete or a male reproductive structure called pollen grain. So when the pollen grains are transferred from here to this stigma, that process is called pollination. And what does that? Most likely insects. So when insects come and perch here, so the pollen grain get attached to their body. Then when they perch here, when they perch on the stigma is very sticky, and the stigma will pick up those pollen grain, and then the pollen grain will move down here to come and fertilize the ovule. So that's pollination. So you will be told to define pollination. So, and one of the agents of pollination for this flower is insect. Next, they ask you, what are the adaptations? What makes that specimen, in this case pride of Barbados flower, to be able to carry out its function? Number one, they have bright colors, petal, so that color attracts insects. Second, the experience of nectar are just like juice which insect comes to suck up. And also they have large surface area for pollen grain reception. So you can see that this part is, the stigma has a large surface area so that it can have the ability to attract or to pick up pollen grain. So moving on is to our next specimen, which is specimen D, is the bean seed, and this is one very common specimen we should be familiar with. So again, for this specimen, you could be told to cut and draw. So this is the bean seed. So you have what called the cotyledon. The cotyledon give rise to the leaf later after germination. So you have the plumo. The plumo later becomes the shoot. Then you have the epicoty, every structure found above the soil. You have the apocoty, you have the radicle. Radicle is later becomes the root. Then you have a little depression. If you look at the bean seed, this depression here is called the helium. So it is through the helium water can pass through or can enter the seed. Then you have a seed coat, this covering of the seed, and that seed coat is called the tester. So now to draw a diagram, because you'll be told to do so. So when after you have drawn, you can simply write a cut section, C-U-T, a cut section of the bean seed. So the next question they will ask you is to identify, or the first question obviously will be to identify, which is simply bean seed. Then the scientific name is called the Vigna unguiculata. Then for the classification rather, you have the plant. It belongs to the plant kingdom. It belongs to the Trachyphyta, and for the class it's a dicotyledon. Then for the observable feature again, what can you see? There's the presence of seed coat, which is also called tester. There's the presence of cotyledon, which is the seed leaf. There's the presence of embryo and other structures you can see like the epicoty, hypocoty, and the helium. So for the uses, what is it used for? It mainly serves as food for man, and is a good source of protein. Also, it can also germinate to produce new plants. Also, the other uses in the sense that it can be used to feed livestock when processed. Now, one good thing is that beans is commonly attacked by the bean weevil, and they can ask you, how do you prevent it, or how can you preserve it, or how can you store it? So one of the ways is, or the ways you can do so is by using of pesticide or insecticide, though it is not ideal, but it can be written on the day of the examination. Next, proper drying of the crop before storage. Next, we can use proper storage facilities. We can also store them in an airtight container. And also, if they are infected one, probably they have been attacked, it is good we destroy them and dispose them of. Next is that you can also handpick. Handpick means you can remove them with your hand. That's the attacking agent. That's the bean weevil, and throw them away. Now, the next question you would definitely be asked is the type of germination. For the bean seed undergo to call an EPG germination. And what is this type of germination? It's a germination where the seed leaf is carried above the soil. So if you look at beans, when beans is growing, for example, you observe that when it germinates, the seed is carried outside of the soil. Unlike maize, when maize is growing, the seed is always within the soil. So a germination where that seed is brought outside of the soil or above the soil is called an EPG germination, and the beans undergoes the EPG germination. So take note, because you will definitely be asked this question. So the next question is the conditions necessary for seed germination. So they are the things that promote germination, and they include sunlight. You need sunlight for germination to take place, water, optimum temperature, oxygen, and good soil. So also they could ask you, what is germination? So germination is simply a stage whereby a seed grows into a what? Young seedling under favorable condition. And it is the condition I've talked about. There must be sunlight, there must be water, there must be good temperature or the right temperature, oxygen, and then good soil. So our next specimen, which is the butterfly, is what you have on your screen right now, and it's a very popular specimen. So this is butterfly. Next, they could tell you to pin the butterfly to the table and draw. So you could be told to draw the dorsal view, which we have here, and this is the lateral view, but you most likely be told to draw the dorsal view because that's way easier to achieve. So for a dorsal view, you have the head, you have an antenna, you have what we call a costal margin, then you have a discal cell, then you have an outer margin, then you have an inner one. So it's for the wing has three parts: a costal margin, an outer margin, and then the inner margin. Then this one above is called the forewings, just like you have the forelimbs in animals. Then this one is called the hind wing. Then you have the abdomen, and that's that for that. Again, the same thing reflects on this lateral view. For a lateral view, you have the forewing, hind wing, the abdomen, the thorax in this case. Also, there's a thorax here. Also, you have the legs, the head, and then the compounder, and then the antenna. Next, the criteria to identify. Again, identify means what can you see? That's the butterfly. That specimen is the butterfly. The next question is the scientific name. The scientific name for the butterfly is called the Danus plexippus. The classification is as follows: the kingdom is Animalia, the phylum is Arthropoda, the class is Insecta because it is an insect. The features, or they could ask you observable features. Number one, it has 4 large and colorful wings, as you can see on the picture I displayed earlier. Then there's the presence of antenna. There's a proboscis. So that proboscis, though not clearly visible here, but it helps it. Okay, it is down here. That proboscis helps it to suck up nectar. Nectars are just juice from flowers. So the proboscis is important for sucking of nectar. So that makes the feeding habit to be sucking. It also have 3 pairs of leg. Let me take you back. So there's one at the front, 1 pair. There's a second pair here, and the third one at the back. So it has 3 pairs of leg. Again, the feeding habit is sucking. The metamorphosis is complete metamorphosis, and I've talked about complete metamorphosis in the sense that it goes from egg to larva to pupa and finally to the adult stage. Next, for the economic importance, like I said, it includes good side of that specimen and the bad side. So for this specimen, it is important for pollination of flowers, and I talked about pollination as the transfer of pollen grain from anther to stigma. Also, the larval stage, which is the caterpillar, is very destructive in the sense that it feeds on the leaves of plants, and when it do so, the plant is unable to grow properly. So that's the bad side, and it also falls under economic importance. Also, they are very colorful, and hence they add beauty to the environment. And lastly, we can use it in biological study. Now, this next question is one good question they will ask you. They will ask you what's the importance of specimen E to specimen C. Specimen E is butterfly. Specimen C is the pride of Barbados. So specimen E acts as an agent of pollination to specimen C. So remember, when we talk about specimen C, I said one of the agents of pollination of C is insect, and one of the insects we can find here is the butterfly. Specimen F is the crayfish, and it's a popular specimen. Now, looking towards this side, this is a picture of the crayfish, and next they could tell you to draw a lateral view of the crayfish. Lateral means a side view. So looking at the crayfish, this is a side view of the crayfish, and it has a cephalothorax. Cephalo means head, thorax again means chest, as you saw in the diagram of a spider. So cephalothorax, the head and the thorax, they are fused together. So this is cephalothorax. Next, you have the abdomen. This is the abdomen, and we have what we call swimmeret or pleopod. It helps the crayfish to swim in water. We have walking legs when it insists to walk with its leg on rocky surface in water bodies. We have what we call cheliped. The cheliped enables it to grab or to tear or for defense. We have antenna for determining the presence, and last we have what we call an antennal, just like the antenna. So moving on, they will tell you to identify specimen F. Identify means what is specimen F, and your simple answer is crayfish. The next is scientific name. Scientific name for the crayfish is the Procambarus clachia. So that is scientific name. And for the classification, it belongs to the animal kingdom. The phylum is Arthropoda, then the class is crustacean. So the features or the observable features, you can see an hard exoskeleton, that's an external skeleton. There's the presence of 10 pairs of walking legs. There's the presence of swimmerate for swimming. We have antenna. And then pincers. The pincers is actually paired for tearing, for grasping, for defense. The next question they will likely ask you is to tell them the habitat. Habitat is the aquatic habitat because it stays in water bodies. But to be more specific, it stays in freshwater. So freshwater habitat are those habitats that have less salt content. So the one that have more salt content is what you call them the marine habitat. So yeah, for the crayfish, it stays in freshwater and the habitat is aquatic habitat. For the economic importance, it is a source of protein. It forms part of people's food and meal. It can also be processed as livestock feed, and also it can serve as a source of employment. For example, those people who fish or who get the crayfish from water, it's a source of employment for them. And also when they do so, it is source of, and when they sell them rather, it's a source of income. They could ask you what are the mode of feeding. So basically the crayfish undergoes an omnivorous, or it's an omnivore, and also a scavenger. Omnivore meaning it can feed on flesh. In this case, it can feed on small fish and other crayfish as well, on tadpole, on insects. And then for scavengers, they can pick up decaying plant or decaying vegetable or decaying water plant, and also they can also feed on other aquatic plants. Next is specimen G, which is the grasshopper, and you mostly find this one in grass areas or grassland. Most times you see them as green, but they can also have other colors. So this is a picture of the grasshopper, and in exam you could be told to draw the lateral view. This is a lateral view of the grasshopper. So for a grasshopper, it has 2 pairs of antennae, it has an head, it has what we call a median carina. This carina helps in providing support and then gives its structure. Then we have the pronotum. The pronotum is just like for protection, helps in protection, just like you have a shield. We have the wings, we have the knee, but this is the, so we have the forelimb, we have the medial legs, and then we have the hind limbs. So for the hind limb, it is long, flexible for jumping or hopping. So we have the knee, we have what we call the femur, we have the tibia, also this is the abdomen. Then we have what we call the fonocule. So this fonocule helps it, or okay, this is femur rather. Then this fonocule helps it in jumping or in hopping. So take note of the various parts of the grasshopper. So we'll move on to the next aspect. So they could tell you to identify. Again, identify simply means the name, which is grasshopper. Then for the scientific name, it is called the Zonoceros variegatus. For the classification, it is under the animal kingdom. Also, it's under the arthropod, and for the class, it belongs to the insecta. For the features of the observable features, that's what you can see. It has long hind limbs, as you can see in the diagram that I showed you earlier. There are 2 pairs of wings. There's a pair of antennae. It has what we call mandible for chewing. Very, very important in your exam. They could ask you the mode of feeding. It is biting and chewing. So it has mandible for chewing. Also, the presence of simple eyes, and that reminds me, the grasshopper has 2 types of eyes. I will call it simple eyes. The simple eyes is for just sensitivity to light. So if it is in a light region, it knows that okay, I'm in a place where there's light. It's just like you close your eyes, but someone point a torch or a light at your eyes, you are able to feel that oh, there's light, but you can't see persons that are walking around you. Then for the compound eyes, it helps to detect movement, that's person's movement around them. So if you are close to a grasshopper, you always notice that the grasshopper will move. Why? Because it knows that, oh, somebody is around. And also it gives a wide field of vision. Then for the economic importance, it's a pest of crop. Take note of that. Also, it also serves as protein source when fed upon by other animals. Specimen H is the humerus of a rabbit. The humerus is the bone found in the arm. So just touch your arm. The bone found in the arm is called humerus. But in this case, the exam body told your school to provide the humerus of a rabbit. So this is a picture of the humerus of a rabbit, and they could try to draw the anterior view. Anterior means the front. Anterior means front. Then they could also try to draw the posterior. Posterior means the back view. So in any case, you should to draw and label. So for the humerus, we'll talk about the anterior first. So the anterior, there's a part above called the greater tuberosity. Then behind, we have what we call lesser tuberosity. Then this is the head of the humerus. Then there's a groove or a little depression here called the bicipital fossa. Then we have a shaft. This is a shaft. Then we have a fossa here called olecranon fossa. Then there's a hole that leads from the front to the back. That hole is called the supratrochlear foramen. Foramen simply means a hole. Then this curved part below is called the trochlea. Then for the back view, just a similar structure with some added advantage or some added features. So again, this is bicipital fossa, just a depression as you can see here. Then we have the greater tuberosity, lesser tuberosity, but at the front we tend to see the head. Then again, this is the shaft. There's a ridge here called the deltoid ridge. You have this coronoid fossa at the back, but at the front you have olecranon fossa. Then again, this is the trochlea, and then the whole is called the supratrochlear foramen. So take note of the anterior view and the posterior view. So they could try to identify simply means what's the name of the specimen. In this case, the name is humerus of a rabbit. Therefore, they would then try to state the scientific name of the organism from which this specimen was gotten from. Now, this specimen, or the one that we provided for you in your school, is gotten from a rabbit. So the scientific name of rabbit is called the Oryctolagus cuniculus. So take note of that. Then next thing we ask, what's the location? The location of this specimen is in the forelimb. So the rabbit has the forelimb, that's the limb at the front. Then the ones at the back, they are called hind limb. But in this case, this is located in the forelimb. Then the classification of the animal, where we got it from. So rabbit is found in the animal kingdom. It is found in the chordata phylum and in the mammalian class or the mammal class. So the next are the features. What are the features, or what are the observable features? So number one, it's a long bone. Second, it is found in the forelimb. Though this is not observable, but they expect to know it. Also, it has a round head. The appearance of a shaft. Now, if they try to talk about observable feature, everything you can see here, you can take them as your answer. So you can write presence of olecranon fossa, presence of greater tuberosity, presence of bicipital fossa. So with all these things, you can write up to 10 observable features of this specimen. So the next question they ask you, what are the functions? So for the function, the humerus is a bone, so it helps in support and movement. Also, it helps in attachment of muscle, tendons and ligament, and also long bones helps in red blood cell production. So it's a very good site for red blood cell production. So they could ask you, which bone is similar in the hind limb? So at the front you have the humerus. So behind, the similar bone to it is what we call the femur, and that is our next specimen. So specimen I is the femur of a rabbit. Again, you have the anterior view, which is the front view, and then you have the posterior view, which is the back view. So for the anterior view, we have the head, we have the greater trochanter, and behind we have the lesser trochanter, and then we have a third trochanter. So in this case, we have three trochanter: greater, lesser, and then a third trochanter. Again, you have the shaft, and then we have what we call the patellar groove and the condyle. In the humerus, we have what we call the trochlea here. So for the posterior, we have the condyles, we have a groove called the intercondylar groove. Why is it called intercondylar? This is a condyle, this is another condyle. Inter means between the condyle. So we have the lesser trochanter, and then we have the head and the greater trochanter. So again, they could ask you the scientific name of the organism from which it was gotten from is the Ornithologus corniculus. But for this case, the location of this specimen is in the hind limb. The humerus is in the forelimb, but this is in the hind limb. The classification still remains the same. And then what are the features? I told you for your observable features, you can always get them from your labels or your diagram. So it is a long bone, which you ought to know. It is found in the hind limb. Then there is presence of a ball. The head is a ball, and this ball helps to fit into the socket joint of the hip to form what we call the hip joint. So there's a shaft. You can even write presence of patellar groove, presence of trochanter, presence of intercondylar groove, and the rest. So moving on, they will ask you the function. The function is just similar to that of the humerus for locomotion or for movement and support, for attachment of muscle, tendon, and ligament, and then for red blood cell production. Then they could ask you what's the similar bone in the forelimb. The similar bone is humerus, which we talked about earlier, or the analog. So they could ask you the homolog, the analog, or the similar bone. So just know that it is humerus in this case. So it means which bone resembles it in position and almost in function. So that's what it means. Then this is a good question. They will definitely ask you to talk about the similarities between specimen, the specimen Femur H and I. So both of them, they are both long bones. They are found in the limbs. They support movement. They are part of the appendicular skeleton. Appendicular means your appendages, and appendages are your limb and your limb girdle. That's your forelimb, your hind limb, and then the girdles. Girdles are what just attach them to the central, the axial skeleton. So just take note, they are both appendicular skeleton, and they both have shaft. You clearly see that in the diagram. Then when they ask you the differences, again, anytime you see a question and the question is telling you to talk about differences, always use a table. So for the humerus is found in the forelimb. The femur is found in the hind limb. For the humerus, it forms the shoulder joint. Then for the femur, it forms the hip joint. Very important. The humerus is shorter, then the femur is longer. Don't forget that. Then the humerus, if you go back to the picture, there was a ridge you saw called the deltoid ridge, but the femur does not have a deltoid ridge. Femur rather has a trochanter, but the humerus does not have a trochanter. So if they ask you this question, just ask yourself, what can you see on the picture of humerus that you can't see on the picture of femur? Or what can you see in the picture of femur that you can't see in the picture of humerus? That will simply form your answer for differences. So what does this one have that the other does not have? Now they could ask you, these two bones, specimen H and I, form which skeleton? They form what we call the appendicular skeleton. As I said earlier, they are your appendages, which include your limbs, your fore and hind limbs, and your limb girdles. So we'll move to the next specimen, which is G, and G is the orange fruit. So orange fruit is a very common fruit, and as you can see, there are two ways they could tell you to draw. They could tell you to draw a transverse section. This is a transverse section, like you cut it sideways. This is a transverse section. So when you cut, you should be able to label. For example, there's the outer part called the epicap, there's the middle part called the mesocap, there's the inner part called the seed. Then you have what we call the seeds. Then there's something that connect the seeds together called the placenta. Then you have a succulent, juicy part. Then again, this is the transverse section, but this is longitudinal. Longitudinal, you are cutting from top to down. So when you cut from top to down, there are some other structures you see, like there's a separating wall or a wall, there's a juicy layer, there's the epicap, mesocap, that's the middle part, and then the endocap. You have the seeds, you have a rind, and then you have the pulp. So those parts you pluck out. So this is a pulp, each of these parts is a pulp. So moving on, we move on to the next part. Again, they could try to identify. Identify simply means the name. Your answer will simply be orange fruit. Then for scientific name, it is the citrus sinensis. Again, I will re-emphasize, the first name should always start with capital letter, second name small letter, and then underline. Then they could ask you classification of this specimen based on their cultural use. Based on their cultural use, it's a fruit. We basically take it as fruit. Then based on the life cycle, it's a perennial. And why is it perennial? Because it grows for more than 2 years. Then they can ask you what are the mode of dispersal. Dispersal simply means how will that fruit leave the parent plant and get to other region. So it's man. When man feed on it, man can throw away the seed anywhere. That's the mode of dispersion. It can also be animals. Animals can also pick up the fruit to a new location. So next type of placenta. It has what we call the exile placentation. So take note of that because you'll definitely be asked this question. Then for the fruit type, for the orange fruit, it's a berry. And a berry is just simply a fleshy fruit. If you look at orange, it's very, very fleshy. It developed from an ovary of a single flower and also contains seed rather, within the fleshy part. Also, the fruit is edible, so that's what makes it a berry. It's just like the tomato. Tomato is also a berry. So think of a fruit that comes from the ovary of a single flower, contains seed and is edible. So that's a berry. Then they could also ask you for the classification. It's a true fruit. True fruit comes from just the ovary only. Then a simple fruit because it comes from just a single ovary. So when it comes from an ovary, true fruit is single ovary, simple fruit. And also it is fleshy and succulent because you can basically eat every part of the orange. Next, ways this specimen can be nutritious to man or add to malnutrition is a source of vitamin. Basically all fruit provide vitamin, that's fruit. It can be used for making fruit juice. It's a source of refuges, and also it can be a source of employment and also source of income for the farmers who are planting and then selling them. The next specimen, which is specimen K, is the honeybee, and it's a very popular insect. So in the exam, you could be told to draw a dorsal view. So here is the dorsal view of specimen K. Dorsal just simply means the back view of that organism. So regardless, don't look at this picture because it is extremely shaded with some inner details. So like I said earlier, just try to draw to your own ability. So it does not necessarily have to be very, very fine. So this is a dorsal view, but this is a lateral view. This one over here is a lateral view. So, but you most likely be told to draw a dorsal view if you have to draw the specimen. Also, for the part of specimen K, we have the antenna, we have the simple eye, which is the ocellus, we have the head. Also, we have a compound eye. So there's a simple eye and then there's a compound eye outside. Simple inside, compound outside. We have the head, we have the foreleg, we have the middle leg and the hind leg. So in this case, it has 3 pairs of leg. You have the forewing and then you have the hind limb. So we have the thorax. This is thorax. This region is the thorax. Then we have the abdomen. Also, there's a pollen comb for picking up pollen grains from flowers, and it is hairy and leathery. So we'll move on to possible questions. So the first question they will most likely ask you to identify, which is what's the name of specimen K, and your answer will simply be honeybee. The next is to tell them the scientific name, which is the Apis mellifera. They could then ask you classification. So it belongs to the animal kingdom. The phylum is Arthropoda, and then the class is Insecta because it's an insect. Therefore, the features, or they could go ahead and say observable features. So it has 3 body parts, namely head, thorax, and abdomen. It has just 1 pair of antenna, 2 pairs of wings, and then if the insect or the specimen is the worker bee, it has what we call a stinger for stinging organisms that are trying to serve as attack, or that are trying to attack it. So that's a stinger. Then for the economic importance, again, economic importance include both the good and the bad. So it's an agent of pollination. Oh, I didn't add the P there, so please, that's agent of pollination. Also, it helps in or it carries out production of honey and wax. So the honey will most times take is gotten from the bee. So that's why it is called the honey bee. So also it's a source of income because people, they do sell the honey. In fact, it's also a source of employment because some persons, they do keep bees for honey production, and when they do so, they can sell them. Next, the habitat is a terrestrial insect. It mainly stays in shrubs, woodland, garden, and then trees. Next, our next specimen, Specimen L, is the land snail. So we have a diagram of the land snail, which is a popular organism. Then we have a diagram down here. So it has a shell. This hard part is the shell. That's this hard part over here. Then you see these lines that are like separating lines. We call them whorls. Then there's a space for breathing, which is called the pneumostome. There's the mantle. There's the head. Under is a foot, or sometimes we can also call it a muscular foot. Then we have the eyes. Then the tentacles. The tentacles hold the eyes in position. Then you have the mouth just beneath, and then lower tentacles. Then next question they could ask you is identify which is the land snail. Then scientific name is the Achatina. Then for classification, it belongs to the animal kingdom. The phylum is Mollusca. That's all your shelled organisms fall under Mollusca. Then the class is Gastropoda. Then for the features, it has a very soft body. If you've touched a snail before, you find out that it has a very soft body. The shell is hard and spiral. It has the tentacles and then a muscular foot. Then what are the uses? It's a good source of food because it provides protein. It serves as employment, especially for farmers. It's a good source of income when they are sold. And then the shell is a good source of protein. That's why most times it is processed and used for feeding livestock so that those organisms can absorb or get calcium and then help them to develop strong bones and other relevant parts where it is needed. So moving on, our next specimen, M, is the guava fruit, and this is a popular specimen, though to some persons, while to others it might just be something rare. So what's the specimen? So you have a picture of it above. Then this is the guava. Then this is a diagram. Again, you could be told to draw in two ways, either in transverse you are cutting it like this, sideways, or longitudinal like this from top to down. So when you do so, you can identify some parts, like there's an outer part called the epicarp, a middle part called the mesocarp. You can just refer back to the diagram for orange. So you have an inner wall called the endocarp. Also there are the seeds and all of that. So next is the scientific name. Scientific name is the Psidium guajava. Again, when you try to identify specimen M, your answer will simply be guava fruit. Then for the classification, based on agricultural uses, it's a fruit. Then based on life cycle, it's a perennial crop. For the mode of dispersal, it's still dispersed by man and animal. For the placentation, just similar to that of orange, it's still the exile placentation. For the fruit type, it's also a berry, just like the orange fruit. Then for the classification, it shares a similar classification like the orange. Is a true fruit because it comes from an ovary, simple fruit, a single ovary. Then it's fleshy and succulent because it has edible and juicy fruit wall. Then the specimens is also a source of protein, can also be used for making fruit juice, a source of roughages, source of employment, and also source of income. Then they will then ask you this particular question: similarities between the orange fruit and the guava fruit. Number one, they are both berries. They both have exile placentation. If you check the placentation I gave earlier, they are both fleshy, they are both edible and rich in vitamins. Also, they are both dispersed by man and animals. Then differences, again, when you are told to differentiate, always draw a table. On one side, place one of the specimen D, on the other side, place the second one. So for orange fruit, it has segment or lobes. Now, most times when some person they eat the orange fruit, they pick them out in segment, like you pick out one, or most times not for orange but other similar citrus food, it has segment and lobes, but for guava it does not have that distinct segment. Then the orange has a very smooth, almost smooth though not fully smooth, but a smooth outer skin, and you can compare to the guava. in which the skin is rough or slightly rough. Then most times when you get close to orange, it has this aroma, which is the citrus aroma, and the inside is very, very juicy. But for the guava fruit, it is less citrus and then more grainy. So the next is the orange fruit, which is orange in color, especially when ripe. Then for the guava fruit, usually green or at some time orange when ripe. Then our next specimen, which is specimen N, is the Irish potato. So this is the potato up here, which we should be familiar with. Then this is a diagram, though you might not be told to draw this diagram, but if they should, just know that you have a proximal end where the stem grows out from. Then you have the eyes, which is best called the lateral bud, as written here, lateral bud. Then you have a leaf scar, which is the point where the leaf falls off. Then you have lenticels, that body. Then you have a distal end. Then they could tell you to identify. Identify means what is the specimen that you can see. Your answer will simply be Irish potato tuber. They will then ask you for the scientific name, which is the Solanum tuberosum. Then for the plant type, it's a stem tuber. Why is it a stem tuber? Because in this case, food is stored in the stem, not in the root. So the Irish potato tuber is a stem tuber. For the features, what can you see? It has a very smooth outer skin. There are numerous lateral buds. So you see numerous lateral bud all across. Then there are presence of leaf scars. And then when you cut it, you tend to see a white flesh. Then for the mode of propagation, it really means how it is planted. It is planted through vegetative propagation, which is an asexual mode of production. So you can plant this tuber, you can cut it off this lateral bud and plant them, and then they will grow to give us another plant. Next, what are the economic importance? It's a very good source of food, that is majorly provide carbohydrates. It is used for provision of industrial starch. Also, it can be used for chips, fries, and also flour production. Also, it's a source of income as well as also a source of employment. So anytime we talk about income, the next thing that should always come to your mind should be employment. Also, it can be exported to other countries for sale. So the last specimen, which is specimen O, is the sweet potato tuber. And this is a picture of the sweet potato tuber, and it's very different in appearance from the Irish potato tuber. So the first question they'll most likely ask you is to identify. So identify simply means what is the name of the specimen you can see, and in this case the name is the sweet potato tuber. Next, they could tell you to write the scientific name, and scientific name for this specimen is the Ipomia patatas. Again, I is in capital letter, B is in small letter, and they are underlined separately. Then for the plant type, it's a root tuber, as against the Irish potato, which is a stem tuber. So in this case, food is stored in the root and not in the stem. Then for the features, it has a taper shape. Taper means a narrowed shape. So it is not like the Irish potato, which you can see here, that has an oval or a round shape. For the sweet potato tuber, it has a tapered shape, or we can also call it the fusiform shape. Then when you cut it open, you'll get to see an orange or yellow flesh within. Then it has lateral bud, or this lateral bud, so it has lateral bud. Then for the mode of propagation, it's propagated vegetatively. That's an asexual mode of reproduction using what we call stem cutting. So in stem cutting, I simply cut off, or I cut these things into smaller parts. Then I use those parts to plant. Then the next thing, the economic importance. So just like the Irish potato, it's a source of food. It's a source of industrial starch. It can be used for production of chips, fries, and flour production. And also, it's a source of employment. And since it's a source of employment, it's also going to be a source of income for farmers. And also, it can be exported to other countries. So in addition, they could tell you to list some similarities and also some differences. For the similarities, they are both enlarged storage organs, in the sense that the organs have been enlarged to store food. For the Irish potato tuber, the stem has been enlarged to store food, in this case carbohydrates. Then for the sweet potato tuber, the root has been enlarged to store food. So in that case also carbohydrate, and that enlarged storage organ is now what we call the tuber. So both of them have rough and irregular surface texture. That's when you touch them, you find out that they are rough. And also they both have lateral bud and also lenticels. Then for the differences, that's the difference between specimen N and O, that's Irish tuber potato and sweet tuber potato. The Irish is a stem tuber, which I've talked about earlier, and the sweet potato tuber is the root tuber. Then for their shape, as you have seen earlier, for this Irish potato tuber, it has a round oval shape. Then for the tip, it has a tapering or a fusiform shape. Then when you cut them open, the Irish tuber potato has a white flesh, then the sweet tuber potato has a yellow to orange flesh. And that brings us to the end of this video lesson on the various specimens for the 2025 biology practical examination. So viewers, I hope you enjoyed the video lesson and you learned something from this particular video lesson. Don't forget to like. Also, do it to drop your questions, comment and suggestion in the comment section of this video, and also do it to share this video lesson to your friends who are writing the 2025 NECO Biology practical examination, so that they also get to learn like you and write the examination in a few days time from now. So if you haven't subscribed to this channel, do it to click on the subscribe button just beneath this video, so that you always get notified whenever I release a new video. And also, when you are done with your examination, do it to come back to this particular video and leave a review for me under this video. So till you get to see my next video, do have a wonderful time.